12:10. Welcome to MVZ Lunch, everyone. I don't have much in the way of announcements other than to remind everyone that next week we don't have an MVZ Lunch. Instead, we'll have this DEI Town Hall, and Carly will be sending out a reminder and announcement about that later this afternoon. Let me just ask if anybody has any announcements they want to make, either by unmuting or in the chat. Okay, then I will turn it over to Carla, who's going to introduce today's speaker.
Thanks, Michael. So it's really a great pleasure to welcome Josh back to the MVZ. Josh was one of our star undergraduates. Graduated in 2013, so almost 8 years ago, which is amazing how time flies. And he went on from Berkeley to work with Craig Moritz, former MVZ director, and also Leo Joseph at the Australian National University, where he got his PhD in 2017. And he already has an impressive 11 publications, some of which came out of his undergraduate work. And he just recently, in I guess January, started a new position at the Center for Biodiversity Discovery at the Museum of Natural Science in Berlin. So we appreciate Josh being willing to give this talk from Berlin. It's 9 o'clock his time. And his job title, which I'd never heard this before, is Researcher Museomics. And so his role is to develop methods for recovering genomic data from museum collections, especially fluid preserved specimens. So it'll be interesting to see how that develops. And anyway, we're so happy to have you back, Josh. And his talk is going to be on comparative genomics of Australian avian suture zones. So welcome back to the MVZ.
Thank you, Carla. Yeah, it was a pleasure to be here. Pleasure to give the MVZ seminar after watching it quite a bit as an undergrad. It's too bad it's not in the Grinnell Miller Library, but at least I'm able to do it sooner rather than later. So let me go ahead and share my screen. All right, make a few adjustments. All right, so the work I'll be talking to you today is about comparative speciation genomics in Australian avian suture zones, and this is really built on the work that I did as a PhD student with Craig, who is also attending here today. And so I'll be talking a lot about the different kind of methods and chapters that I'm working on, as well as some of the stuff I've been developing even after my PhD. And if there's time, how that kind of builds upon some of the research I want to be doing in the future and the program I want to build. And so to start things off, when I talk about speciation, I'm really talking about kind of this divergence between a more kind of phenotypic and genetic or genomic the homogeneous population into fully diverged isolated species and all the stages in between throughout that process. And when I talk about this, I'm talking about kind of this variation within, and I'm referring to this kind of term, the speciation continuum, and how populations, or one might call species, kind of move between this gradients between one population to two species, either being pushed onto one side by mutation, selection, and drift, or getting homogenized by gene flow and recombination. And what's important to note is that this is not kind of a one-directional path, and species, or being a species, is not necessarily an endpoint, and you really have this kind of movements between these other ends due to these different forces and different kind of contexts in the environment. And so when we study speciation, so the hybrid zones really lend itself to a lot of these studies. So it serves kind of as this natural experiment to test this degree of reproductive isolation. And so when we look at hybrid zones, we can actually kind of map out and see this kind of variation in phenotype through space, placed in the ecological context of the environment that these kind of populations and species are evolving in. And so with genomic tools, we can start to look at, okay, what's going on in the kind of genomes of these birds or whatever organisms we want to study in the hybrid zones. So we can start to map out ancestry through space, across the environments, and then start to get into these minute details, such as introgression through the genome. So one of the jobs, though, that we have as kind of speciation biologists is start to tease apart these different things that make this studies quite complicated. So you have lots of different sources of selections, lots of different genomic features that affect this pattern that we see across the landscape, as well as the pattern that we see across the genome, such as recombination rate, gene density, and so on, placed in the context of the demographic history that these populations or species are experiencing, as well as the geographic context that they are placed in. And so it's really difficult to really kind of tease all these things apart. So we try to focus in on some of these things at a time. And so here are just some of the kind of research that's been done in hybrid zones and speciation, and kind of a variety of things that we started to learn. So as Michael was saying, Jochen came over almost 2 years ago now, a year and a half ago. So he was talking about the hybrid zones in crows, and studying that, we were able to get at understanding kind of the phenotypic integrity. How can you have this kind of rampant gene flow within the genome and yet maintain these very divergent phenotypes across space? You have the classic case of adaptive introgression, the heliconius butterflies, where certain loci would kind of introgress through. So then you kind of adopt the mimicry. You can also use this to start mapping incompatibilities within the genome, seeing which actual parts of the genome are responsible for the isolation between the species or the population, as well as starting to look at chromosomal rearrangements and how that affects kind of divergence and speciation at different levels. And so if hybrid zones are kind of this natural experiment, I'd argue that suture zones would be really quite useful in understanding a lot of the questions that we have. So suture zones, for those of you not familiar, are hybrid zones that are clustering in geographic space. Initially, were defined by Remington in 1968. So this definition, though, was expanded a little bit further by including contact zones and phylogeographic breaks also clustering throughout geographic space. And so what we're really getting at are these populations or species pairs, which then share this climatic history as well as geography. And at the same time, we start to get these pairs with different degrees of connectivity as well as different degrees of reproductive isolation. So the goal is that to be able to start looking at these kind of different stages of speciation in a natural setting. And so how do suture zones form? So here are just some of the main hypotheses that we have is formation of the suture zones. So first is the refusal expansion. So if they were isolated in shared areas and expanded about the same time, then they would meet in and around the same areas, shared by geographical barriers, or you can have them kind of meeting towards foothills, or sometimes that would also happen between kind of lowland species as well as more montane species. And for plants, it's more common to get this happening due to anthropogenic disturbance. And so what are some of the advantages studying the suture zones? As I mentioned before, you have shared climatic history, shared biogeographic context, and convenient field sampling. So you can just go to the field and sample multiple individuals from different species and hybrid zones at the same time. So what can we learn from hybrid zones? So here we can start to get at how different species or populations respond to similar contexts, how the different movements of genes across geographic space occurs in these different contexts, as well as this variation of degree in reproductive isolation in these different populations. And so, like I was saying before, if these hybrid zones serve as a very kind of useful natural experiment to test this degree of reproductive isolation, then suture zones kind of serve as replicates of these natural experiments. Of course, it's not necessarily perfect, but it's really good at kind of getting at these populations with different levels of divergence and isolation and shared geographic and climatic history. And so I'd like to share just some of the kind of projects that have been done with the suture zone work and speciation. Though not a lot has been done just yet, and I'm hoping that this is something that will pick up pretty soon. So here are just a couple of projects from Benjamin Winger and Sonal and Kay, who are also at the MVZ. So this first project is getting at phenotypic divergence and genome divergence in birds in South America. So what Benjamin did is compare these kind of 16 co-distributed lineages. So you would see some of the sampling here on the figure. It's my pointer. I'll just use the arrow, hopefully you guys see that. And kind of compare within 8 different genera how the superspecies and the monotypic comparisons work. So you have these kind of multiple comparisons, and he used ddRAD SNPs to compare the genomic divergence as well as phenotypic divergence within the genera, as well as between or among them. And so what we found is that there's this kind of correlation with phenotypic divergence associated with this genomic divergence. And this divergence is also associated with much lower gene flow. And so then he's arguing that there's this relevance of the phenotypic divergence with speciation in this particular system, which is a bit of a contrast to other systems, especially the one I'll be talking about next. So this study by Sonal, kind of stemming from her PhD with Craig as well, is looking at a lot of cryptic hybrid zones in the wet tropics in Australia. And so she was looking at these 4 different contact zones, also at different levels of divergence, and then use pool-seq and exon capture to kind of compare parental populations as well as the decline that's kind of observed between them. And what she found was this kind of clustering, or this kind of congealing of the genome. And how to define this a little bit better is that you're kind of just looking at, so these are the kind of decline widths of the different loci within the genome, and the light blue is less diverged populations, and the red ones are much more diverged. And so you just see how much it kind of narrows down, the different widths of the different slips in the genome narrows down, and how far it deviates from the center on this right panel. And this is kind of lending this to this idea of congealing or snowballing of divergence in the genome during the speciation process. And so what we're really getting at when we're asking these questions is starting to go from genes to genomes to populations to species, try to understand all of these different dynamics and how it relates to the speciation process. So before I get started. All right, so here's a bit of a structure and how my talk's going to be. I'll initially talk about how I established this comparative system and start to then move on to asking some of the different questions and how the role of geography and divergence in the speciation process. And if there's time, I'll try to kind of touch upon a little bit of the work I was doing as a postdoc and some of the future prospects I have for this project and some of the ideas. And some of the stuff I'll be presenting to you today is a bit preliminary, so it'll be really useful if I think quite a few of you guys in the audience might have a bit of insight in this. So it'll be great to have a bit of a discussion afterwards and what I'm doing wrong or how I can improve some of these analyses. Start off with a comparative system, asking how does genetic and phenotypic divergence vary in the suture zone. And once we get into the role of geography, looking into range connectivity, divergence, and gene flow, how gene flow decreases once you have increasing divergence or increasing isolation, how hybrid zone width correlates with this divergence, and kind of what evolutionary history is consistent with the patterns that we observe in these suture zones. To start things off, Australia, avian suture zones. So yeah, Australia is a great kind of system for this because a lot of these avian suture zones have already been mapped out by Joel Craycraft in 1986. So the dashed lines you see here are the kind of transition zones, or he wasn't calling it suture zones at the time, transition zones of these birds where you see a transition between one population or one, say, phenotypic morph or one subspecies transitioning to another. And so there's been quite a few that he's already been mapped out. So it's really quite convenient because it can just pick out from these systems what to study. And so with that, okay, how did these transition zones form? So you have these different, to start it off, you have these different areas of endemism. And then a lot of these areas also have a lot of climate refugia. So it kind of alludes to some of the stuff I was talking about before with how suture zones form, as well as a lot of shared biogeographical barriers in these regions. And so what that results in is ecological divergence, periods of isolation and secondary contacts, and then a lot of barriers to gene flow, both current and past, of course. And so getting into the system I chose to study here was the Melophagidae. So this includes a few families of birds, so including fairy wrens, including pardalotes or the Australian warblers, as well as the honey eaters. And the main reason why I chose this for this type of kind of comparative study is that there's a lot of species. It's easier to kind of go to one region, choose a subset of these species, and then you kind of have your replicates for these comparative studies that we'll be talking about later on. And so first off, how does genetic and phenotypic divergence vary in a suture zone? So with this, initially looked at some fields, some samples we already had in the museum, and it's kind of compared in this suture zone in northeastern Australia. So just did some we just looked at some of the individuals from Cape York, some kind of contact zone between and eastern Queensland, and used 35 species that are all co-distributed in this area. So did sequence capture as well as mitochondrial DNA sequencing to kind of just scope out what's going on in this. And then I also pair this with some of the morphometric measurements that's been done previously in some of the museum skins. So really, this is kind of just a pilot project trying to check out what's going on in this suture zone. And so a lot of you guys are probably not strangers to the haplotype network. For those who are not too familiar, each of the dots, the colors in the dots represent a different geographic region. The size of these circles reflect how many individuals there are. And the hashes in between them reflect kind of genetic steps. So here, I'm just showing you the mitochondrial ND2. So we're really just looking at this one gene. And the main thing I want you guys to take out of this particular kind of busy plot is that there's really quite a bit of variation where you have some of these birds really quite mixing with these different populations, while others have really kind of strong divergence and strong population structure. And so these are all co-distributed. They have quite similar sizes, similar ecologies, and you still see quite a bit of variation in terms of their population structure and divergence. And so once we start to look at the morphological divergence or pair this up with the morphological divergence, so what we see is that first off, like I said, we have quite a bit of genetic divergence varying across the suture zone. And we also have quite a bit of morphological divergence. Though we might get a bit of a pattern coming out in the mitochondria, we don't really pick this up in the autosomal and the Z chromosome. So this is in contrast to what Ben saw in his study before. And maybe if I chose some more divergent individuals or divergent species, kind of beyond the kind of sister species range, we might pick up a little bit more. But what I want to take out of this is not necessarily the discoveries that he had is not necessarily consistent with all the systems we might look into. And so with that, I now have 35 different samples or different systems that I can sample for these later questions. So the first question I have is, how does range connectivity affect divergence and gene flow? So with this, I'm really getting at the classic geographic modes of speciation. And what I mean by that is comparing this kind of allopatry, parapatry, and sympatry. And kind of looking more in the range dynamics. So allopatric, there's no overlap in the ranges. Parapatric, you have a bit of overlap, and sympatry, you have full overlap. And how that's necessarily relevant to speciation is really the roles that these different kind of mechanisms or these play that help drive this divergence between populations, as well as homogenize it. So in allopatric, you have a lot of genetic drift really kind of coming into play. Gene flow is not really kind of playing much of a role here. And divergent selection doesn't necessarily need to be super strong. And on the other end of the spectrum, in the sympatric speciation, it's a lot of gene flow. You need a lot of divergent selection. And while in parapatry, you kind of have a little bit of everything playing a role, from genetic drift, gene flow, and divergent selection. And so in our system, we're not really looking at parapatric speciation. So here I'm really kind of emphasize allopatry. Sorry, I'm not looking at sympatric, but so I'm trying to emphasize allopatry and parapatry. And so how does this kind of break down during the speciation process? So here we start off with our ancestor population. So they're all kind of gray, they're all similar. Early stages of divergence, you start to accumulate some mutations that are different between these two lineages. In our parapatric scenario, you still have kind of high gene flow going on between them because you don't have a lot of kind of reproductive isolating mechanisms accumulating just yet. Through time, with later divergence, this gene flow might be a little bit decreased, it's moderate, you still have a bit of movement. While in an allopatric situation, it kind of still keeps going and diverging. In the end, once we have full species, allopatry is how you expect it. In the parapatry, you might have a little bit of gene flow still kind of going on, but it could also complete with no gene flow left over. And so kind of getting at that, I kind of zoom out a little bit and start to include samples in Papua New Guinea. And this really serves as our kind of allopatric control, or allopatric comparison. So here I compared 4 populations: Northern Territory, Papua New Guinea, Cape York, and Eastern Queensland, and sampled a few individuals in each of these populations. So I only took a subset of the 35 that I mentioned previously. So here I just took 8, and these are 8 that are all co-distributed in these regions. And I also kind of specifically looked at these ones that vary in kind of degrees of divergence. So then we can really try to get at how these contexts affect the different levels of divergence among these comparisons. And the analyses around here, so I collected DD-RAD loci and looked at population divergence, and then used species distribution modeling to look at connectivity, and then help project this in the past to see how that kind of historic connectivity was occurring in these populations. So kind of first off, just quick kind of networks of populations from the DD-RAD loci. And this is really just to give you an idea that you have quite a bit of variation in terms of structure. So the way you want to look at this is that if the colors reflect the different geographic regions, and if they're kind of clustering together, and that means they are more related to each other, as opposed to ones that are kind of much more intermixed. It means there's not much population structure going on. And the grays, the kind of gray blobs around them, just reflect kind of predefined either species or subspecies taxonomically. So it's kind of underlaid in the genomics here. Like I said, what you see is quite a bit of variation in both population structure as well as divergence in the nuclear, kind of reflecting the mitochondria that I showed in the previous slide. And so what does that actually mean in our parapatry allopatry comparison? So what we see here, so this is just looking at both the differentiation or divergence in the genome, as well as a metric for gene flow, which I'll expand on a little bit further. On the x-axis, we're going to look at pairwise comparisons of these populations with increasing distance between the populations. So the one on the right will have more distance between them as opposed to the ones on the left. And the colors reflect whether or not current geography shows that they're currently allopatric or they're currently parapatric, which means that there's connectivity between them. And I think what I want you to take out of this is that you don't really see much kind of variation between the allopatric and the parapatric comparisons. You might expect the genetic differentiation to be higher in these allopatric comparisons, but that's not really what we see here, where in our parapatric comparison, you actually get a lot more divergence. And the same pattern occurs in the gene flow. This is a little bit inverse though. So the top will have the highest probability of gene flow. So you have most gene flow happening when the populations are at the very top, as opposed to the ones in the bottom. So these are kind of more complete, more isolated, not a lot of gene flow is occurring between these populations. So you see more of a pattern with increasing distance, but you don't really pick up any pattern between the blue and the orange in the allopatry and parapatry. So here I'd argue that the current allopatry and parapatry doesn't really correlate with divergence in gene flow. And so once I look at the species distribution modeling and what I project kind of in the past, and start to correlate this genetic differentiation with the time point where they had the least amount of resistance. So it's kind of getting at their connectivity in the past, or how much opportunity for gene flow that they were able to experience, or then you see a kind of clearer correlation. So rather than kind of looking at these contemporary ranges, it was much better to get this kind of pattern once we look at their past connectivity. And so what does that actually mean for our allopatric, parapatric definitions? Well, I'd argue that these systems is not really quite that simple, that we can define these based on what we see today. And particularly for this system and probably a lot of other systems, you have more of a dynamic climate and dynamic connectivity, where you have kind of pulses of gene flow during the past and then periods of isolation, rather than kind of just something continuous throughout time. So then you have varying influence of selection, varying effects of drift, and varying effects of gene flow as these populations diverge and accumulate reproductive isolation. So how does range connectivity affect gene flow and divergence? historic connectivity rather than contemporary geography is a better predictor. And I guess it might seem a bit obvious now, but I'm glad to be able to test it out in this particular system. And so getting at the next question, how rapidly does gene flow decrease with increasing divergence? And so here is something I'll kind of explain a little bit further. So here I'm looking at this kind of expectation of the likelihood of gene flow and divergence. So I'll explain the likelihood of gene flow a little bit further in the next slide, but what you want to see here is the parapatric kind of starts off at 1. So you can think of this as kind of a composite of both the kind of isolation, repetitive isolation occurring, as well as the opportunity for migration between these populations. So the allopatric, if they're allopatric, there's no opportunity for migration, so that kind of remains at 0. It's likelihood of gene flow as the populations diverge, as opposed to the parapatric populations, where you have kind of high likelihood of gene flow early on, but as it accumulates these kind of isolating mechanisms through time, you get this kind of transition to lower likelihoods of gene flow until speciation has completed, or it's really low that you don't have a lot of gene flow going on anymore. And like I said, you have this kind of dynamic movement between mutation, selection, and drift and gene flow as these kind of populations move between either side. So that's kind of just getting at that. And this point at which this kind of orange transitions is kind of mostly complete reproductive isolation. But I said it doesn't necessarily mean it's going to stay there, and it could kind of move back depending on how environments change. And so this uses that same system that I mentioned previously, but here I'm kind of incorporating some of the ABC analysis and demographic modeling. And so to break this down a little bit further, so I'm kind of just comparing these models without migration and models with migration. I'm kind of lumping all of these different scenarios within. So without migration, you have strict allopatry, ancient migration, and with heterogeneous kind of effective population within the genome. So that's really to reflect kind of variation in recombination rates and effects of linked selection. And with migration, you have isolation of migration, secondary contact, and all the variations within. And the main reason why I'm kind of lumping this together is that our capability of teasing apart these different scenarios really depend on how much divergence has occurred, how much time these kind of populations have been separated, and how long it's been since they came together. And so depending on the system you're looking at, it may be easier or more difficult to tease them apart. So if I want to compare these different systems, it's not necessarily going to be fair if I want to kind of tease these apart. So I think it'll be much more straightforward just to kind of lump them all together without migration and with migration. And so within this, when I'm talking about this kind of probability of gene flow, so we have our kind of simulated data sets of these different scenarios, and then our empirical data sets. And so really what I'm trying to get at with this likelihood of gene flow is that if our empirical data set, if all the simulated data sets close to it all have kind of models of supportive gene flow, it'll be at 1. So it's 100% of the models will have it. If none of them have gene flow, it'll be 0. And then everything in between, depending on how many of the models have gene flow or without. And so going back to our little plot, or the kind of expectation that I showed you earlier, so you have this allopatry where you kind of expect throughout divergence, you don't really have likelihood of gene flow going on. And this parapatry, you have this kind of transition point. And so once I map the empirical data onto this, keep it gray for the time being, what you see is none of them are really following this kind of allopatric trajectory. And all of them are kind of following this transition of parapatry. And once we color them in, there's no obvious kind of pattern that we see over here either. And so with that, try to get at, okay, how quickly does this actually accumulate? How does this transition occur? And so I'm just comparing these different models and trying to see, okay, which of these patterns really reflect the empirical data that I showed you previously. So here are 5 different models proposed. So threshold is just a quick transition from one not being a species to being full species. Constant is kind of this constant accumulation of reproductive isolation. Accelerating means you have this kind of rapid rates in the beginning and then immediate completion. deceleration, though here it looks kind of similar to constant, though this should really reflect kind of the opposite of the acceleration. You have a slow rate in the beginning and then an immediate completion. And sigmoidal is kind of have the slow start, rapid middle, and then a slow end. And so really it's kind of mapping, seeing which ones of these models are most consistent with the data that we have. And so with this, the best models that are supported are actually the accelerating and sigmoidal. And it's a bit difficult to kind of tease these two apart because below, there's just not a lot of data. So I think the one with the highest support is actually accelerating, with sigmoidal being quite close. So I kind of like left them a bit together and leave that up to interpretation. And so like I was saying before, this kind of support for gene flow on the y-axis. And then on the x-axis, you have this kind of level of divergence. And here I'm using FST as a proxy. And main reason why I'm doing that is just I want it to be limited between 0 and 1. So if we use a different kind of metric of divergence, such as EXY, that might doesn't really bound that in this kind of limits. So like I said, the best supported models here is kind of with a slow start and rapid accumulation. But whether or not this kind of completion is much slower, it's not too conclusive with the data set that I have here. So last couple of questions, how does that hybrid zone width correlate with divergence? So here we go back to sampling different systems from that 35 that I mentioned previously. And instead of sampling allopatric populations here, we did more of a two-dimensional transect across the landscape, and then also used DE-RAD for this particular analysis. So here are the 10 kind of candidates with different levels of divergence like previously, and then I analyze population divergence, admixture, and incline widths in the system. Start things off, here's kind of NGS admix, or the highest level amounts of hybridization. And what I want you to get from this is that you just get a variation of introgression. And so those of you who are not too familiar with reading this, each kind of bar, each horizontal bar reflects a single individual. So the ones with more bars, or the species with more bars, would reflect species with more samples. And then the color reflects kind of their purity of the more northern population in red, southern population in yellow, and those kind of in between would have different levels of admixture between them. So this shows the extent of hybridization of the different systems. And we really see quite a bit of variation here. And so as admixture proportions, I use these admixture proportions to get at Klein width in between. And so those of you familiar with this, Klein width is correlated with dispersal distance over the square root of selection. And though we normally look at Klein widths in kind of a SNP by SNP basis, I don't really have that kind of resolution in this sampling. So I'm just using this admixture proportion for our Klein width. And since looking at some ecological data, dispersal distances between these different systems tend to be quite constant. So really Klein width in this context should reflect selection. And so then I look at divergence of the allopatric populations to see which ones are not currently being affected by gene flow, although that might have been affected by gene flow in the past, but already kind of completely homogenized. What we get is this pattern. So on the x-axis, we have divergence. On the left is FST, so differentiation in the nuclear genome. And on the right plot is the mitochondrial P distance. And then on the y-axis, we have hybrid zone width. So higher up, wider, lower down, more narrow. And so what we get here in our 10 systems is that we see this really rapid kind of transition to much narrower hybrid zones, just with kind of meager changes in divergence. And the same pattern holds for both ND2 as well as kind of genomic or autosomal divergence. And so what that means, or how I kind of would interpret this, is that a lot of the neutral loci could have already introgressed in the past, so that would have already been homogenized. So what we have with higher divergence, we might be able to start to think about this as potential signals for higher reproductive isolation. So instead, we have this higher divergence also associated with more kind of genome-wide restriction in gene flows. It's also more restricted in geography. So the more diverged the populations are, the more geographically restricted the gene flow tends to be, which is what we see with these narrower hybrid zones. And so how does this correlate with divergence? Hybrid zone width is drastically reduced with a marginal increase of divergence. And so the last question we have here is which evolutionary history is consistent with the patterns in the suture zone? So this is the preliminary work I was talking about that I'd really appreciate a bit of insight on. So here we use the same system, same 10 kind of systems that I was mentioning previously, same two-dimensional transect. Really, it's kind of just complementing the pattern that we see previously with some kind of simulations of different evolutionary scenarios, trying to get at, okay, which scenarios could have formed or could have resulted in the patterns that we see today. So here are just some of the scenarios I tested, or the main ones I've tested so far. So what could have caused this variation and divergence in gene flow in these suture zones? First, is the different times in secondary contact. So maybe they didn't come together at the same time like we would have expected. So you have varying time to the point that they're reaching equilibrium. Second is different degrees of contact. So maybe some of these are experiencing more of a kind of harder barrier compared to the others. So then that means that even though they came together at the same time and been isolated at the same time, they don't really move across the landscape similarly. And so that might be resulting in this variation in gene flow and divergence. And lastly, it's a different time in allopatry, which also reflects both varying differences in neutral divergence and varying number of incompatibilities. And I'll get at that a little bit once we explain the model. And so this is a little bit more kind of what we expect in the suture zone scenario. So you might have variation in the kind of number of incompatibilities. And so let me try to break down these simulations a little bit further. So I'll try to keep it simple. 36 populations, simple stepping stone model. So you have kind of just movements between your neighboring populations in this stepping stone. So we have these two populations, the blue and the red. There's no ecological difference between them in these simulations. And I keep population size, sequence length, combination rate, mutation rate, and so on, and incompatibility consistent across them. And so the only thing that I add on is this kind of incompatibilities. And this is really post-zygotic incompatibilities. So to break this down a little bit further, if an incompatibility appears in our blue population, it functions as if it was neutral, just fine until it is present in the genome with incompatibility that arose in the pink population or the red population. And the more incompatibilities there are, the lower the fitness of the hybrids tend to be. And so with these models, I mean, I won't be presenting on all these different variables today, but I also changed the migration rate, time in allopatry, secondary contact, connectivity, and tested whether or not we can get these patterns in purely neutral versus scenarios with incompatibilities. And so how I did these simulations, so you had your ancestor population, let it evolve for quite a few generations, period of allopatry, so it's really kind of just restricting the gene flow between the red and the blue populations. And the reason why I kept it like this is that you can still kind of accumulate isolation by distance in these systems, which is something that we should expect when we're looking at the geographic patterns. And then afterwards, secondary contact. And then after secondary contact, I would sample these kind of populations and see what the dynamics of gene flow and divergence are and how that kind of changes what we observe. And so, like I said, comparing this back in sort of empirical data, what I showed you previously was this FSC and allopatric and how that relates to hybrid zone width. And then what we see on the other side is that how FST and allopatric populations is reflected in the contact zone population. So the first one is different times in secondary contact. So how to break this down a little bit further is that the variation within a panel is this kind of metric that we're looking at, is different times in secondary contact. But between panels are just different contexts. So here, the different columns reflect different times since allopatry, though all of them share the same type of migration rate. So what you want to do is compare kind of the empirical panels on the left with the simulations on the right to see how similar they would be. The red dot, so each of these dots are pairwise comparisons and they're kind of whole genome comparisons of our population. So it really reflects what I was showing you in all the previous plots. So the red ones are situations or scenarios where there are some loci that are or have incompatibilities, though this of course includes all of the neutral loci as well. So we don't know exactly which loci are incompatible or not, but we know that they have incompatibilities in there. And the ones that are gray are completely neutral. There are no incompatibilities in them at all. And so what we see here is that the simulations don't really reflect what we see in the empirical data. Maybe a little bit, but not too much in either way. So you have more of this kind of linear association with the FSC in the contact zone and allopatric, but it's not what we see if the variation within or between our populations is due to different times since secondary contact. So we might be able to rule out that they kind of came back together at different times in the past. So the next is different degrees of contact. So it's like I was mentioning, once they come back into secondary contact, maybe some of them would have a stronger barrier than the others. And I mean, I think it's quite clear here that it also doesn't reflect what we might observe in our empirical data. It's more of a kind of linear relationship where the allopatric FST kind of stays consistent among all of our comparisons, but what really changes is FST in the contact zone as well as the hybrid zone width. But the allopatry doesn't change too rapidly. But once we get to the different times in allopatry, which also, like I said, reflects different degrees of incompatibility accumulated, we see a much more kind of consistent pattern with our empirical data. So you have a more kind of linear relationship with a FST in the contact zone and FST in the allopatric populations, as well as this kind of rapid decrease, like I mentioned previously, of the hybrid zone width in the FST in the allopatric populations. And so which evolutionary history is consistent with the patterns in the suture zone? At least with the comparisons that I've done so far, I would say it's different times in isolation, and the amount of incompatibilities is the most consistent in the patterns that we see. Like I said, this is quite preliminary, so yeah, I'd really appreciate some kind of feedback on this. One of the things I really want to change soon, one of the next things I want to kind of try out is actually changing this hybrid fitness dynamic into more of a kind of snowballing, like I mentioned previously. So you have kind of slowed down at first, rapid accumulation of, or a rapid decrease of fitness, I guess in this case, as you have more incompatibilities accumulating. And so let's review this questions. How does genetic phenotypic divergence vary in a suture zone? Both of them vary, though they're not necessarily correlated. Range connectivity, historic is probably much better, predictive of gene flow and divergence rather than contemporary connectivity. How rapidly does gene flow decrease? If you have a slow start, then a rapid transition. And hybrid zone width correlate with divergence. Higher divergence is associated with this rapid decrease in hybrid zone width. And this is more kind of reflecting of a history of varying amounts of either isolation or number of incompatibilities. And briefly with some of the future prospects, I really want to take this, so the comparisons I was showing you today was mostly just ddRAD and kind of whole genome comparisons. I want to use the genome that I assembled with a fairy wren as well as some of the genomes kind of in the pipeline. So I'm working on a few PacBio hi-fi genomes and Hi-C genomes and whole genome resequencing to try to see some of the patterns and variations that we see in the whole genomes actually within the genome itself. And then kind of lending that to some of the work that I've been doing with Jochen is that really trying to understand how to infer recombination rate variation within the genome. And kind of pairing these two together, what I'm really interested in is kind of observing this kind of variation in genomic space, as well as geographic space in a comparative spatial context. So really trying to get at speciation in these different perspectives, so then not just necessarily pairwise comparisons of populations, but across the geographic landscape, as well as different levels of divergence. And with that, I'd like to thank you for listening and open up for questions.
Well, thanks, Josh, for a great talk. I'm sure Josh will entertain any questions people have.
Josh, I have a question. That was a lovely talk. When you were comparing the probability of gene flow with genetic divergence from DD-RAD data. How did you deal with, I'm sure you've thought about this, but the circularity of inferring those two things from the same set of data? So you had FST versus the probability of gene flow. And presumably under many situations as FST increases, as the genetic variation is partitioned in such a way that FST increases, you will necessarily infer less gene flow.
Yeah, so with that, I was really kind of looking into the simulations. So whether or not we can actually recover situations where there are lower FST and still quite low gene flow, and whether or not what I infer is an artifact of my data or is it an artifact of something else. And I didn't show that information here today, but just looking at the simulations which use the same data to infer both divergence and gene flow, there should be scenarios where there is really low divergence and allopatry and no gene flow, but we didn't really recover that in the empirical data itself.
Josh, I had a quick question. Really nice talk, thank you. So I was curious, I don't know if you have the data for this yet, but I know that there's a fair amount of plumage variation across some of the taxa that you looked at across the contact zone. So I was wondering if you had data or you could speculate on where you have taxa in parapatry versus allopatry, where the chromosome rearrangements are playing a greater or lesser role in helping, shape the curve of how you get that isolation.
Yeah, that's a good question.
If you've got any insight, because it's still so unexplored in birds. We all assume there's such synteny, but the most radical phenotypes are often inversions.
Yeah, I mean, unfortunately, I do not have that data yet. So, I mean, in terms of the plumage variation, there is like you said, there's quite a bit of variation. Though with my data, it's not particularly correlated. So a lot of the ones with higher divergence tend to also be quite cryptic. They're kind of just brown and very different. Versus some of the ones that do have quite a bit of phenotypic divergence, there's still quite a bit of gene flow. So in that aspect, it's not necessarily similar to in this system. But I'd really like to get into the chromosomal inversions eventually once we have the data for that. Not quite yet.
Josh, can you say anything about vocal divergence in this system and these different
That's a good question. I never got around to that, unfortunately. I wanted to. Like going out in the field, there was nothing particularly obvious to me. But we really just got closer to the contact zone a little bit more in the northern end of the southern populations. And so, once you get a little bit further down, maybe, but it's not something that's really been explored to my knowledge.
I'll just ask one more question if I'm allowed. So within the habitat mosaic that separates the north and the Cape York and the Queensland, did you, when you looked at the contact zone, did you see any correlation with habitat type, like woodland or more open habitat type related to the width of the contact zone, given the number of pairs that you had?
Yeah, definitely. So this is why I wanted to get at this, at the very end, this kind of connectivity. So the more woodland birds tend to have much wider ranges versus the ones that are kind of associated with rainforest as well as mangrove tend to be more narrow. Of course, they tend to be much closer to the coast themselves. And so they experience quite a bit. I think they might be experiencing different levels of kind of connectivity in that sense. And I was wondering whether or not that was affecting this dynamic, which at least with these simulations didn't show that it tends to be. But yes, there is quite a bit of variation depending on the habitat.
I wonder if I can come in with a question from afar. Hello, everybody. It's lovely to see you all.
Hey, Greg.
Great talk, Chelsea. I really enjoyed it.
Thanks, Greg.
Actually, I had a different question, but Rory's question prompts me to think about the population structure of those different habitat types. So your simulation drew a linear stepping stone, which reductions in gene flow are exaggerated compared to two-dimensional stepping stones. We've known that since Maruyama in the 70s. And so the woodland birds are essentially two-dimensional systems. but the rainforest and mangrove ones are essentially linear systems. So I wonder if that different arrangement of the different habitat types might also influence the outcomes in your relative to your simulations.
Yeah, that was one of the things I wanted to get at next with this. It was just this was a bit simpler for the time being, so I started off with that. And especially because like I think kind of the connectivity with a two-dimensional kind of lattice is a little bit more complicated, but I do want to get at that and see how that affects things. But I do think it would.
Yeah, but I think the simulations are really cool, and you started in a logical place with them for sure. My other question is, I've got excited recently about population haplotyping, high throughput population haplotyping, and of course that's right between the postdoc you did with Joachim and you know, your ongoing work on this system. How do you think that might change how we can study hybrid zones of speciation and adaptation and so forth?
Oof, that's a good question. No, I think it would change a lot, and I'm really excited to kind of get at that. Yeah, I'm really trying to push to do this kind of haplotagging in the museum here, but it's going to be a bit of an effort, but I think it's definitely doable, and it's quite affordable. It's going to be cheaper, about the same price as a standard library prep. So hopefully in the future we can get these kind of haplotypes. But I think it really changes this dynamic because, well, one, I think we start to be able to get more out of these individuals or like smaller sampling once we actually get at haplotype blocks and how that kind of breaks, rather than trying to sample, say, 30 individuals per spot. So it kind of, the classical clinal analysis, we're looking at that. So we kind of start to learn, okay, how does this break? Does it break based off of what it expects just due to recombination and neutrality, and whether or not it starts to break down much further in? And so I think we start to also get at timing of secondary contact a little bit further once we complement that with some of the recombination information, as well as whether or not some regions are more kind of restricted due to selection, or there are some kind of adaptive integration occurring here. So I think there are a lot of capabilities once we get into the haplotyping world.
Cool. Thanks.
So Josh, related to that, this is a question I always get asked, so I'm going to ask you it.
Oh no.
So that with the recombination rate that you mentioned, but also for haplotyping and other things, you know, it always comes back to what you're going to use to estimate that rate. So do you have an idea of how you could estimate it for honey eaters or something like that, instead of having to use chicken or Ficedula?
That's a very question. It's a very difficult one, to be completely honest. So the work I was doing with Jochen was, and I think he mentioned this during his talk, was that I was trying to get at kind of male recombination rate using bird sperm, which is definitely doable, but of course it has its limitations because, one, it's not that easy to get at sperm on all species, and two, you're really only getting at male recombination rates rather than an average of the female, which we know is kind of really related to the divergence that we see in these populations. And so, at least to start off, I would probably look at a different population that's not influenced by, say, gene flow in the context of these species, to infer more of the kind of landscape of divergence using LD hat or LD helmet. And I know that is quite limited, but I think one way to kind of get around the circularity is to use an independent species, or different independent population, sorry, not species, so that it's not really kind of influenced by these different dynamics in your hybrid zone or contact zone, because those different dynamics, as well as, you know, expansion and contraction, would start affecting this landscape. It's, you know, it's not the perfect workaround just yet, and it's something I think hopefully will get a little bit better in the future.
Hi, Josh. Nice to see you. I wanted to ask, are you continuing all this work with your new job, and sort of what's involved in being a museumomicist, or whatever what your title is? That sounds amazing. So, and are you going to work in other taxa, like some herps or something else too?
Yeah, that's a good question. So tied to this is this position's, when people ask me exactly what I'm doing, I'm also trying to figure this out. So the first kind of primary objective of this position is to figure out how to extract DNA from museums using specimens. So a lot of it tends to be invertebrates and insects. But on the side, I'm also kind of establishing my own research group. And so with that, this is really what I want to focus on in terms of comparative biogeography in the context of speciation and kind of combining variation in the landscape of the genome as well as the geographic landscape. But of course, it's something I really want to start expanding both geographically as well as different taxa. So I'm quite open to different people and different projects to see what we can start to learn in these comparative types of analyses. So yeah, it's kind of these two sides. So they'll have the museomics on one side as well and have this comparative biogeography and speciation on the other.
Any other questions? Okay, well, let's unmute and give Josh a round of applause for.
Yay. Great job, Josh.
Josh,
Sure.
see you.